The ear is responsible for translating variations in air pressure, whether from speech, music, or other sources. The ear can be divided into three principal sections: the outer ear, middle ear, and the inner ear. Each of these parts performs a specific function in processing sound information. Sound waves are first collected by the outer ear, which consists of the external ear and a canal that leads to the tympanic membrane, also known as the eardrum. The configuration of the external ear amplifies sound, which is important for speech perception. The shape of the external ear is also important for sound localization. From the ear canal, the sound waves vibrate the eardrum, which in turn vibrates three tiny bones in the middle ear: the malleus, the incus, and the stapes. The stapes vibrates a small membrane at the base of the cochlea in the oval window, which transmits amplified vibrational energy to the fluids of the cochlea. The round window separates the tympanic canal from the middle ear. The complex structures in the inner ear convert sound into neural activity. In mammals, the auditory portion of the inner ear is a coiled structure called the cochlea. The region nearest the oval window membrane is the base of the spiral. The other end, or top, is referred to as the apex. Along the length of the cochlea are three parallel canals: the tympanic canal, the vestibular canal, and the middle canal. The endolymph is the fluid that is in the membranous labyrinth, and the perilymph is the fluid between the bone and the membrane labyrinth of the ear. The principal elements for converting sound into neural activity are found on the basilar membrane, a flexible structure that separates the tympanic canal from the middle canal. The cochlea narrows toward the apex, which vibrates in response to sound transmitted to the fluid by deflections of the oval window initiated by the bone of the middle ear. Within the middle canal and atop the basilar membrane is the organ of the chordae, the collective term for all the elements involved in the transduction of sound. The organ of chordae includes three main structures: the sensory cells, which are hair cells, an elaborate framework of supporting cells, and the terminations of the auditory nerve fibers. Atop the organ of the chordae is the tectoral membrane, and the stereocellular membrane of the outer hair cells extends into indentations in the bottom of the tectoral membrane. The movements of the fluid in the cochlea produce vibrations of the basilar membrane. These vibrations send the stereocilia inserted into the tectoral membrane. Depending on the direction of the bend, the hair cells will either increase or decrease the firing rates of auditory nerve fibers. Voltage is the gated calcium ion channel that controls the amount of calcium allowed in. Potassium ions are necessary for the function of all living cells and can be found in the inner ear. Depolarization also occurs in the inner ear, which is a change in a cell's membrane potential and removes polarity. The neurotransmitter is any of the chemical substances that transmit nerve impulses across a synapse to a postsynaptic element. Exocytosis is the transport of materials out of a cell, such as a sac or a vessel, that first engulfs the material and then extrudes it through an opening in the cell membrane. Finally, the vestibulocochlear nerve is either one of the 8 pairs of cranial nerves that supply the cochlea and the semicircular canals of the internal ear, which contribute to the sense of hearing. Thank you.